it's my great pleasure to introduce Olga Schubert, who is going to give today's talk. Olga has been a terrific postdoc fellow in the lab. She's done some really exciting work on the genetic basis of variation in how much of a given protein the cell makes, and has done some really amazing and pioneering technology development to enable her to do that, that I think will have quite broad applications even beyond the specific very interesting topic. She's also been a real sort of glue person who's held the lab together in many ways, and has just been a real pleasure to have. And she's gonna move back home to Switzerland this summer to start a position there, and we're gonna really miss her. So Olga, take it away.
Okay, thank you very much, Leonid, for the introduction, and thanks to Caroline for the organization. Let me share my screen. Okay, so yeah, I guess many of us here try to understand in some way or another how the genome sequence affects the phenotype of an organism. However, as we all know, this is not a trivial task because there's so many processes involved in getting from a genetic sequence to a phenotype. So I guess in order to reach that ultimate goal of one day being able to know or at least predict the functional role of each individual base pair in a genome, I think it will be very useful to look at some intermediate molecular phenotype. For example, the proteome. And I'm focusing on proteins because cell physiology is mostly determined by proteins. They serve as structural components. They are the enzymes catalyzing reactions, the constituents of signaling networks from receptors to transcription factors. So as a molecular class, proteins really reflect the cellular phenotype more directly and more comprehensively than mRNAs or metabolites, for example. So the overall question I'm interested in is, what are the genetic factors determining protein abundance? And more specifically, I'm interested in things like how many genetic loci affect the abundance of a protein. What fraction of genetic loci have specific versus broad effects? What are the underlying molecular mechanisms and things like that? So we're approaching this question by systematically introducing genetic perturbations across the genome and map out their effects on the abundances of individual proteins. And we're doing this work in the yeast Saccharomyces cerevisiae because it is a tractable eukaryotic model system with a relatively compact genome. And as a tool to introduce these genetic perturbations in a targeted fashion, I'm using the CRISPR base editor. The base editor consists of an impaired Cas9 that can still be programmed with a guide RNA, but that instead of cutting the DNA, introduces C to T point mutations in a highly specific targeted fashion guided by the guide RNA. And the particular base editor that I'm working with, base editor version 3 or BE3, has been developed and characterized in mammalian cells. So we first needed to see how well it would work in our yeast cells. So for this, first we had to develop a plasmid system that would allow us to express the base editor together with the guide RNA in yeast cells. And we then set up a pilot experiment with 10 guide RNAs. First, we induced the base editing process, and then we deep sequenced the genomic target region for each of the 10 guide RNAs to see whether we can detect the expected base edits. And this figure here shows an overlay of the 10 targeted genomic regions aligned by the PAM site. And this gray box here indicates the guide RNA target region. And you can see nicely here that the base editor introduces only C to T point mutations in a defined window, 13 to 17 base pairs upstream of the PAM site. And when we look at the efficiency, we find that for about half of the guide RNAs that we tested, the base editor works with high efficiency, editing 50% or more of all the cells in the culture. And for over 3/4 of all the guide RNAs that we tested, the base editor edits at least 20% of all the cells in the culture. So overall, the editing profile and efficiency of the base editor in yeast is highly predictable and also actually very comparable to what has been described in mammalian cells. So having this established, now let's look at how we can use the base editor in a screen to identify genetic loci that affect protein abundance. So here we are starting with a culture of identical cells in which one gene of interest is tagged with a GFP. And then we transform these cells with the base editor and a plasmid-based guide RNA library. And some of the mutations that are introduced by the base editor in these guide RNAs might then lead to changes in the levels of our protein of interest, as will be reflected by changes in the GFP signal. We can then isolate the cells with the highest and lowest GFP levels using flow cytometry, and then sequence the guide RNA plasmid in the selected populations to find out which guide RNAs are enriched. And as is the case for all CRISPR screens, each of these enriched guide RNAs then obviously reports on a particular mutation that affects our protein of interest. So our guide RNA library consists of 16,500 guide RNAs that together target a total of 4,500 genes out of the 6,000 genes annotated in KIST. And the library is composed of the following 3 types of guide RNAs. We have a third of the library that introduces early stop codons in non-essential genes. So these are basically gene knockouts. And then we have 2 sets of guide RNAs that target conserved residues in non-essential and in essential genes. So overall, this means that all guide RNAs that we are working with introduce coding mutations with potentially large effects on the target genes. Importantly, this screen has to be repeated for every protein of interest, each time starting with a different GFP strain. So for the dataset that I will be presenting here, I did this screen 11 times for 11 different proteins. And these proteins include multiple proteins involved in core cellular functions, like key enzymes in glycolysis, fatty acid synthesis, histone, a ribosomal protein, and an enzyme involved in DNTP synthesis and DNA replication. We also included 2 proteins involved in stress responses and 3 GAPDH isoenzymes, which I will talk about in more detail later in the talk. So this is an overview of all the hits identified in our screens for the 11 proteins, which you can see here in yellow. In total, out of the 16,500 guide RNAs that we tested, about 1,000 guide RNAs had a significant effect on at least 1 protein. And these 1,000 guide RNAs target 694 genes, and that is actually what is shown over here in this network. So every node here represents one of these 694 genes whose perturbation has a significant effect on the abundance of 1 or more of these 11 proteins. On the outside are the genes that affect only 1 of the 11 proteins when perturbed, and on the inside are those that affect 2 or more proteins. So in our screen, on average, the abundance of a protein is affected by 155 genes, and over half of these genes affect multiple proteins. So factoring in that we did not target the genome exhaustively and that not all guide RNAs work efficiently, this suggests that most proteins in yeast are affected by hundreds of genes, and that most genes affect the levels of hundreds of proteins when they are perturbed. So in short, the genome-wide protein regulatory network is very highly interconnected. Now, when we look more closely at the directionality of the protein abundance changes, we see that most of our genetic perturbations reduce protein levels reflected here by these purple bars, whereas we have fewer genetic perturbations that increase protein levels, as shown here in orange. But interestingly, this pattern differs somewhat depending on the functional role of the protein. So proteins involved in core cellular functions mostly decrease, so those over here, whereas proteins in stress responses equally frequently increase in response to genetic perturbations. This is again the same graph as before, but each column split by target gene essentiality. So the fact that we see bigger red bars than green bars means that the perturbations of essential genes are more likely to affect protein levels than perturbations of non-essential genes. And what this figure also shows us is that the predominantly negative regulation of protein levels that we've seen in the previous figure is mostly driven by perturbations of essential genes, whereas perturbations in non-essential genes affect protein levels in both directions. So overall, perturbations of essential genes are more likely to affect and reduce protein levels than perturbations of non-essential genes. Now, when we look at the number of proteins that are affected by each gene perturbations, which is what I show here on the horizontal axis, we find a strong overrepresentation of essential gene perturbations among the perturbations that affect many proteins. So 8 or more here of the 11 proteins that we studied. And in contrast, non-essential gene perturbations in green tend to have no or much more specific effects on protein levels. So overall, we can conclude from this that perturbations of essential genes are more likely to affect a larger number of proteins. A big question here, and in general for our screens, is that are these effects on protein levels direct or indirect? So on the one hand, gene perturbations can directly affect protein levels, for example, through the mutation of a regulator of that protein. But of course, gene perturbations could also indirectly lead to changes of protein levels, for example, by affecting the growth rate, which would in turn trigger secondary adaptations in the cell. So this is something that we cannot directly address with the data from our screen, but we do have indications that both these factors, direct and indirect, affect protein levels. Now let's briefly come back to this network overview of all the 694 perturbations that with a significant effect on at least one of the proteins. So as briefly mentioned earlier, about half of all gene perturbations affect only one of the 11 proteins on the outside, whereas the other half on the inside affect 2 or more of the proteins. And this is exactly what is also shown in this pie chart here, which reflects the number of proteins affected by each gene perturbation. And what we can see here nicely is that some gene perturbations affect a really large fraction of our 11 proteins. For example, up here, we have 29 gene perturbations that affect 8 or more of the 11 proteins. And interestingly, when we look at the functional annotations of these genes, we find that the two most enriched GO terms here are ribosome biogenesis and tRNA metabolism. So two functions clearly related to protein biosynthesis. In contrast, for gene perturbations with more specific effects, the most enriched GO term is transcription by RNA polymerase II. So this suggests that perturbing genes involved in transcriptional processes or regulation often lead to specific protein changes, whereas perturbing genes involved in post-transcriptional processes can lead to very broad changes in protein levels. Now, I would like to take a closer look at these genetic perturbations up here with the very, very broad effects. So this here is a small protein-protein interaction network of those 29 genes. And as we've seen on the previous slide, most of them are involved in ribosome biogenesis and tRNA metabolism. And again, we don't see perturbations of genes here involved in transcriptional processes. And maybe one possibility could be that cells are just better able to compensate for broad transcriptional perturbations than for post-transcriptional perturbations. And there's actually a study that specifically suggests this. So now I would like to highlight one of these broad regulators, that's POP1. POP1 is an essential gene that is involved in ribosomal and tRNA maturation. And I just want to note here that because POP1 is an essential gene, we wouldn't have been able to identify it with a normal CRISPR-Cas9 or a transposon screen, for example, because a complete knockout of POP1 wouldn't be viable, so it wouldn't be recoverable with a screen. But because the base editor allows us to introduce partial loss of function mutations, we can identify also essential protein regulators as well. So one reason why POP1 caught our eye is that in studies of natural variants in yeast, the genomic region around POP1 was also identified as affecting many proteins. But the causal gene in these studies has never been identified because this genomic region encompasses like almost 60 candidate genes. And we now think that it is quite plausible that POP1 is the causal gene underlying these regulatory hotspots in wild yeast strains. But it's something that still has to be validated. Now, POP1, as well as most of these 29 genes upon perturbation, lead to lower protein levels, which makes sense given their involvement in protein biosynthetic processes. But as you can see, there are a few exceptions down here. And for example, that's CYT4 and SUB155. So CYT4 is a PP2A-like protein phosphatase that is involved in various cellular functions, and SUB155 is a protein that is strictly required for the function of CYT4. So it makes sense that these two proteins, these two genes, when perturbed, have similar effects on protein levels. CYT actually stands for suppressor of initiation of transcription. So that would actually suggests that it's a transcriptional regulator. However, it has also been linked to the ubiquitin proteasome system, which would again be a post-transcriptional function. In either case, the perturbation of a regulator with either of these negative or suppressive functions is indeed expected to result in increased protein levels. So that is consistent. Now, for the last part of the talk, I would like to shift gears and discuss more specific regulatory mechanisms of individual proteins that we discovered. So across the 11 screens for the 11 proteins, we found many known regulatory relationships. For example, we found regulators of histone gene transcription that affect the levels of our histone when perturbed. Or we found components of the TOR signaling pathway, including two important transcription factors that affect the levels of our ribosomal protein when perturbed. So a lot of things that are either known or totally make sense. But we also found numerous yet unknown regulators, and I would like to walk you through some examples involving the three GAPDH isoenzymes, TDH1, 2, and 3. So GAPDH is best known as a key enzyme in glycolysis, but has also been implicated in many non-metabolic processes in mammalian cells. For example, histone gene regulation, protection of telomeric DNA, apoptosis, stress responses, and so on. In yeast, however, these non-glycolytic functions of GAPDH are much less understood. And interestingly, yeast has 3 isoenzymes for GAPDH, not just one, like most other organisms. So in yeast, TDH3 is thought to be the main enzyme for GAPDH function in glycolysis, but the role of TDH1 and 2 is much less clear. So the one thing we know is that they are upregulated in certain conditions, but the regulatory mechanisms and the functional relevance of that are unknown. So that's why I thought it would be interesting to include all 3 of them in the base editor screens and see if we get some clues here. Here are scatter plots to pairwise compare the regulators of these isoenzymes. And each dot reflects a genetic perturbation with a significant effect on the 2 proteins that are being compared. What we can immediately see here, for example, is that almost all regulators that are shared between TDH2 and TDH3 affect them very similarly. But now when we look at regulators shared between TDH1 and 2 and 1 and 3, we see that some regulators have opposite effects on these proteins as well. So let's layer on some functional annotations to better understand this. Now we can see that many of the shared regulators are involved in central carbon metabolism, suggesting that all 3 isoenzymes might have some role in glycolysis after all. But let's also look at some more specific regulators. For example, here are 2 regulators that affect only TDH1 and 2, but not TDH3. These are SSN3 CDK8 and SRB8 MID12, 2 subunits of the regulatory CDK8 module of mediator. Both of these genes have been implicated in carbon catabolite repression, which is a mechanism to suppress genes involved in the use of alternative carbon sources, as long as one of the preferred carbon sources, so mostly glucose, is available. So seeing that TDH1 and 2, but not TDH3, may be under control of carbon catabolite repression suggests that TDH3 is responsible probably for glycolytic flux under standard high glucose conditions, and TDH1 and 2 played a role when glucose is scarce. Another set of regulators with an interesting pattern here are those that affect TDH1 and 2 in completely opposite ways. So strikingly, most of these are members of the RAS-PKA pathway. The RAS-PKA pathway is a core pathway in yeast and one of the main drivers of cell growth and proliferation in response to nutrients, and in particular glucose. So I colored here all the members of the pathway that we identified as regulators for TDH1 and 2. And the activating components are in red and the inhibitory components in blue. Now, this heat map, which is also grouped by activators and inhibitors, shows very clearly that all perturbations of activators that lead to increased TDH1, lead to increased TDH1, but decreased TDH2 levels, whereas perturbations of the negative regulators of the pathway have the opposite effect. And we confirmed these findings also by a completely independent set of methods using mass spectrometry-based proteomics. So overall, this data reveals a new link between the RAS-PKA pathway and the 3-GAPDH isoenzymes, suggesting that TDH1 and 2 play distinct roles in different cellular growth states or stress responses that are regulated by PKA. In conclusion, for this last part, we found that TDH1, 2, and 3 are very distinctly regulated, clearly suggesting functional diversification of the 3-GAPDH isoenzymes. And I think especially this last part of the talk serves as a good reminder on how much biology there still is to discover with these types of genetic screens, even involving key regulatory pathways in very well-studied model organisms like yeast. And with this, I would already like to conclude my talk. So to quickly summarize, we developed a high-resolution genetic screen using the CRISPR base editor to study the genetics of protein abundance. We uncovered general features of how protein levels are affected by genetic perturbations, but also identified many new regulatory relationships. So I hope that through future studies with denser mutagenesis and more protein readouts, we can further refine and complete our understanding of the genetic factors influencing protein abundances, and that basically see this work as a first step, opening up a huge space of possible extensions. My overall vision would be to eventually build a comprehensive quantitative model to mechanistically explain and predict the role of each genomic position in gene and protein regulation, and then hopefully link that also to various cellular and organismal phenotypes. I would like to thank all the members of the Krugliak Lab for their contributions and providing a great atmosphere in the lab and discussions. And in particular, I would like to thank my advisor, Leonid, my collaborator and friend, Josh Bloom. And yeah, thank you for your attention, for coming, and I'm happy to take questions if there are any.